All right, so today we'll be talking about qPCR, qualitative polymerization chain reaction. So in this presentation, we'll first talk about the purpose of qPCR. So basically what it is and what we use it for. And then after that, we'll talk about the steps within qPCR. So those steps will include first extracting the RNA from our sample, and then converting those RNA that we extracted into cDNA. And in our second part of the presentation, we'll talk about why we'll be converting into cDNA, as well as how we extract RNA from the sample. And then afterwards, we will talk about how qPCR itself works. So that includes basically doing the plating for qPCR, as well as how the qPCR machine works. And then lastly, we'll talk about the basics on how to interpret the results that is provided after the experiments. So we'll first go into the purpose of qPCR. So why should we use it? So before we dive into that, we'll just do a quick review on what central dogma is. So within cells, the genetic information within cells is known as DNA. So how cells can pass on their DNA and don't lose their DNA over time when they replicate is that they have something called DNA replication. So they do this continuously as they multiply themselves so that the next generation of cells would have the same DNA as their parents. And then alongside with having DNA, they need to make that DNA useful. So what they need to make is something called RNA. And the process of going from DNA to RNA is called transcription. And then if you were to go backwards from RNA back to DNA, will be called reverse transcription. And RNA is just an intermediate molecule that allows protein to be made. So once RNA is made, there will be something called ribosome that translates the RNA into proteins. So that is what central dogma is. Central dogma describes the 3 steps of how DNA is being replicated, as well as how DNA becomes protein, which is useful in cells. So one question is, thinking about central dogma, if we're measuring the amount of RNA, what would we be measuring directly? So if we measure the amount of RNA that is inside the cell, what does it show that is happening inside the cell? So we're going to what is qPCR. So qPCR is a real-time reaction that basically measures the amount of the target DNA sequence that's being converted into RNA. And then how do we measure that RNA? Is that we monitor the amount of amplification needed for that amount of RNA to reach threshold. So there's a defined threshold that we measure, and we'll be seeing how many cycles it takes for that amount of RNA to be amplified to a certain amount and passes the threshold. So, and how we amplify RNA is through the polymerase chain reaction. All right, so there are 2 different ways of how we use qPCR. So qPCR can be used for diagnosis or detection. So basically seeing whether a gene is present inside the DNA. For example, with COVID-19 testing, we can amplify a patient's DNA and see if there is any viral genome inside that patient's DNA, and therefore we know whether that person has been infected by COVID-19. The second part will be gene expression analysis. So we're trying to measure the relative amounts of gene that's inside the DNA. So one is looking at whether the gene is present, and the other way is looking at how much of that gene is inside a sample's DNA. So for our experiment currently, so we're trying to use qPCR to determine the relative changes in gene expression levels of the key neuropeptides between fruit flies of different sex, so male, female, virgins, as well as gene knockdown. So certain genes are being knocked down in some flies, and we're using that to compare with other flies without the gene being knocked down. All right, so we'll move on to the steps in qPCR. So the first 2 steps would be RNA extraction as well as cDNA conversion. So we'll be discussing why we need to do these steps for qPCR. So the first step for extracting RNA in our sample is something called homogenization. So the purpose of homogenization is to create a uniform and consistent mixture of the sample cellular components. So basically, what happens is that, for example, in a fruit fly, all the cellular components are inside the fly, and we want to open the fly up so that its cellular components will be exposed so that we can easily access those components and measure them. So how we do homogenization, so we'll use something called homogenizer beads. So these beads are being added to the test tube where the fruit fly is sitting, and when we spin those beads with the fruit fly around using a homogenizer, the beads will break open the fruit fly and universally disperse the cellular components in the sample inside the test tube. And how the homogenizer works is that it vigorously shakes the tube with the beads in there to break the sample open, as well as distributing those cellular components evenly throughout the solution. And then we have something called centrifuge. So centrifuge, what it does is that it separates the substances. So after we do the homogenization where everything that's inside the fruit fly is being exposed, we want to separate those substances inside that solution according to particle size and density differences. So different particles inside the fruit fly will have different density and different sizes, and we just want to separate those particles so that we can distinguish different cellular components. For example, this layer will be nucleic acid, other layers will be protein, lipids, etc. All right, so now we get into the RNA extraction in fruit flies. So one of the main problems that we have would be our ability to isolate RNA in fruit flies without damaging the RNA that we're going to measure. And we also don't want the RNA to be contaminated with other biological products, such as DNA, proteins, lipids. So our solution to that problem would be using something called trisol. So why we use trisol is that it has the ability to maintain RNA integrity without disrupting the cells and dissolving cell components. So what trisol does is that when we basically break open the fruit fly, homogenize its components, we want trisol to keep the RNA safe and intact so that we can measure them later on. However, we still want the cells as well as other cell components to be dissolved and basically disrupted. So there are 4 reasons why we use trisol for tissue homogenization. So the first reason is that it has low pH, acidic. So due to its acidity, it allows the separation of RNA from DNA and protein. So since we're only measuring RNA in our sample, we don't want RNA to be mixed together with DNA and protein, and acidity would be a really good way to separate these molecules apart. The second reason is that trisol contains something called guanidinium salts. So it has its ability to denature the proteins by weakening the hydrophobic structure. So it's something called chaototropic agents. So it will denature the protein, and it will be useful when we're trying to separate the proteins that are attached to RNA. The third thing is that within trisol, there's something called phenol. So phenol is an organic compound, and it can be used to nucleic acids and proteins. And we'll be discussing more about why organic compounds are important for RNA extraction later on. And then lastly, it prevents the activity of RNase. So RNase are enzymes that break down RNA, and we don't want our samples to be broken down. So trisol also has this ability to inhibit these RNase enzyme to protect our RNA sample. All right, so we'll go into our first concept, something called phase separation. Phase separation is the process of separating organic compound from its aqueous solution by shaking or mixing with a suitable solvent. So it usually results in having 2 phases, known as organic phase and aqueous phase. So we'll see how those works. So in order to do phase separation, we need to centrifuge the sample. So when we centrifuge our sample, it does two things. So the first thing would be phase separation, which we talked just recently. So what it does is that it separates the two phase based on some compounds could be an aqueous, and then some compounds will be in the organic phase. And then by putting them in the centrifuge, we also separate them based on the density and particle size. So we'll see how it happens in our next slide. And then for the second thing that happens in centrifugation is something called precipitation. So in addition to separating molecules based on the density and particle size, it also exerts more gravitational force than what you're experiencing on Earth. And that would bring together the precipitate, so basically solid molecules that are coming out of the liquid phase into solid particles, and to help these solid particles to settle at the bottom of the solution. And that'll be really useful for our experiments as well. All right, so this is our game plan to isolate the RNA. So if you just remember recently, we talked about how phase separation requires a suitable solvent in order for it to happen. That suitable solvent would be something called chloroform. Chloroform is an organic compound, and it separates biological molecules and solvents based on their water solubility. So chloroform is an organic compound, so that means usually water-soluble molecules won't be able to dissolve in chloroform. So therefore, it allows the separation of two layers, the aqueous and the organic layers. So once we add chloroform in our sample, we're going to vortex it as well as centrifuge it. And what results is that you get something called phase separation. So as you can see two distinguished layers on the left-hand tube where we'll be extracting one of these layers out that contains RNA. So the left-hand tube is the tube containing the head sample, and the right-hand tube will be the tube containing the body sample. So you can see that the layers that are produced look very different from one another, and the colors are also very different too. So the reason why the head has a more dark brown color at the top, as well as the pink color at the bottom. So usually the head contains more red pigments on. Okay, there are more red pigments on the head, so therefore it will look less clear compared to the body for the top layer. And then the second layer, it contains what's in triazole, so phenol, and that is also pink. So having the red pigments from the sample's head, as well as the pink pigments from the triazole, when they mix together, they get the second layer here. And then for body, they usually have a less amount of red pigments, so therefore the phase separation that is produced is not as reddish compared to the head sample. All right, so after we have this phase separation and we took out the first layer containing the RNA, we're going to extract those layer, and we're going to put isopropanol in that RNA fluids. So what isopropanol does is that it significantly lowers the solubility of RNA in the solution. So it basically forces those RNA to precipitate out of the aqueous solution that it is in. And then afterwards, we'll also vortex and centrifuge it. And the last step of isolating RNA is that we'll wash our extracted RNA pellets with ethanol. So it basically get rid of other aqueous contaminants that are stuck in the RNA pellets, so that what we have left will be pure RNA. All right, so we just talked about centrifuge. Does anyone want to answer why we centrifuge in between these steps? Why do we vortex solutions? Two reasons in those two steps. So after the step of adding chloroform, we want to promote the binding of those chloroform with other organic molecules, so water insoluble molecules, so that they all go into the organic phase. We don't want any organic molecules going to the aqueous phase. So that's why we want to mix it really well, so that the chloroform, which is an organic compound, will bind to other organic compound that's inside the sample and basically push them all towards the aqueous phase. And the second reason, after adding isopropanol, is that we want to promote the RNA precipitation in the entire solution. So we just add the isopropanol inside the solution. It might just sit at the top or sit at the bottom based on the density, and then not all of the RNA inside the test tube will precipitate out. All right, so we'll talk about, this is for phase separation. So there are 3 phases, as you've seen when you do your experiments. So the top layer, which is a clear layer, is known as the aqueous phase. So that contains the RNA. And in the middle, there's that white cloudy layer that is known as interphase, and it contains DNA and proteins. And then the bottom layer is known as the organic phase, usually looks pink, and that would contain chloroform, phenol, and lipids. So organic phase contains all the molecules that are not water soluble, while aqueous phase contains molecules that are water soluble. All right, so if you remember from our game plan, which phase should we pipette out in step 2? So this is a summary with RNA extraction. So triazole, what it does is it protects the RNA while we want to break apart the sample itself. So we add in triazole, and then we homogenize the entire test tube. So we break open that fruit fly while protecting the RNA, but then we kind of want to have everything else exposed. So once we homogenize the test tube with triazole, we want to add in chloroform. Chloroform, what does it do? So it's trying to promote phase separation would be the goal. But it is true. It tries to get all those organic compounds inside the test tube to go in the organic layer. So after that, we want to vortex it and incubate and also centrifuge it. So then we get our phase separation. We get those 3 layers. And then we'll want to extract out the aqueous layer, which is the top layer that contains RNA. And then we want to transfer the aqueous layer into a different test tube. And we add isopropanol in to facilitate precipitation of that RNA out of the solution. And in addition to that, we also vortex it, centrifuge it, and then we want to remove all the excess liquid. So after the RNA precipitates out, you'll see that white pellet sitting at the bottom of the test tube. So that means that everything else, so the liquidy part, they're not RNA. So you want to get rid of that. And once you remove the supernatants, all you have left is the RNA pellets sitting at the bottom of the test tube. And afterwards, you add ethanol to purify, get rid of any excess aqueous contaminants in there. And then we will add RNase-free water in there to resuspend the RNA pellets. And we also use heat in addition to water for the resuspension of RNA pellets as well. So in the review step, we talked about resuspending the RNA pellets back into RNase-free water. So the reason why we want to do that is because we want to look at the amounts of RNA inside and the purity of those RNA using UV spectroscopy. So what UV spectroscopy provides is a value known as A260 to 80. So what it is the absorbance of a diluted RNA sample measured at 260 and 280 nanometers of light. So the Biot-Lambert law predicts a linear change in absorbance with RNA concentration. So the higher amount of RNA concentration that is in our sample shows that would show a higher amount of absorbance, and we're measuring that absorbance using the UV spectroscopy. All right, so as I mentioned, the absorbance have a linear relationship with the RNA concentration. So when we put that sample in the UV spectroscopy, we'll be able to determine how much RNA is in our sample, so how much RNA we collected from what we just did with the RNA extraction. And it is also important because we need 1 micrograms of RNA in qPCR steps later on. So by knowing the RNA concentration, we can calculate the volume of that solution to extract that 1 micrograms of RNA. And secondly, it also determines the RNA purity. So if we see any abnormal values of A360 to 80, then it shows that our RNA sample might be contaminated with other substances, for example, residue phenol, guanidine, and other reagents. So the ideal RNA concentration reading for 260 to 280 would be around 2, and for 260 to 230 would be around 1.8 to 2. So if we see any values that are significantly out of range, that might show that our sample might not be as pure as it is. All right, so the second part of doing qPCR is to convert RNA into cDNA. So 2 reasons why we do that. So first, we use DNA polymerase at the qPCR step to basically amplify the DNA that's in our sample in order to see how much of those cDNA in there. And for the DNA polymerase enzyme, they only recognize DNA rather than RNA, so we cannot use DNA polymerase to polymerize RNA. So therefore, we need to convert RNA into cDNA before we amplify them with this enzyme. The second reason is due to stability. So cDNA is a lot more stable and more convenient to work with. And RNA is very easily degraded by any omnipresent RNase. So RNase is almost everywhere. So you need to, for example, use RNase-free soap to wipe your working area to make sure that there's no RNase present to break down your RNA sample. All right, so there are other benefits. So the one that we use to convert RNA into cDNA is something called iScript-gDNA Clear Kit. So there are some benefits to it. So it is very special because first, inside that kit, it has something that can digest gDNA. gDNA is genome DNA. So there's something that can digest that, and then we do that before we convert RNA into cDNA. So does anyone have any guesses of why we want to remove genome DNA? So here's some hints. So you get a test tube, supposedly containing RNA, and then you get some gDNA in there. And do we want gDNA in our RNA sample? Yeah, so because we'll be converting the RNA into cDNA later, we don't want any pre-existing gDNA in our sample because those will also be measured later on with the qPCR step. So we only want to measure the amount of cDNA that was being converted from RNA, and not any gDNA that was present before we start that conversion. And then the step 2 about converting from RNA into cDNA, that's performed in the PCR machine. So we'll go through about reverse transcription. So reverse transcription is how we convert RNA into cDNA. So first you have a single-stranded RNA sequence that we collected from our fruit fly sample, and then we'll use primers. So primers are something that will initiate the conversion. So initiate basically a polymerization chain reaction. So it will bind to the basically poly A tail. So at the end of the RNA sequence, there are just a tail of adenine base pairs. So you have a primer of thymine to bind to that, and that will initiate the reverse transcription. And then once the primers bind to that, then the reverse transcriptase can start putting in the building blocks, and the building blocks would be basically different base pairs. So cytidine will bind to guanine, and uracil will bind to adenine, etc. And it just keep going until for the entire RNA sequence. And because uracil is binding to adenine, and uracil is only specific for RNA sequence and it's not found in DNA sequence, so what is produced would be basically it's complementary DNA. So for the priming of the RNA strand, so for these little primers to bind to the RNA strand, it will happen in 25 degrees Celsius for 5 minutes. And then for the reverse transcriptase to start polymerizing the RNA strand into cDNA, it would happen in 26 degrees for 20 minutes. All right, so you can see that the reverse transcriptase continue to put more base pairs to match the base pairs on the RNA. And then once it's finished, so you get a single-stranded cDNA, and this will be what we have after reverse transcription. So we stop the reverse transcriptase from continue polymerizing at 95 degrees, and it only lasts 1 minute. And then after we obtain this complementary DNA, we can just keep that DNA stable at 4 degrees temperature. All right, so this is what is in the iScript reverse transcription supermix. So this is what we use for reverse transcription to happen. So inside this transcription supermix, we have reverse transcriptase. So reverse transcriptase is the enzyme that converts RNA into cDNA. And then we also have something called RNase inhibitor, and that prevents the breakdown of RNA in your sample while reverse transcription is happening. And then we get something known as deoxynucleotide triphosphate, so also known as DNTP if you've seen that in the diagram before. So what it is just basically building blocks that the mini base pairs that you see that the reverse transcriptase use to form its complementary base pair on that RNA strand. So that is basically the building block of the complementary DNA. And then afterwards, we also have buffers. So buffers can maintain the pH of that reaction, and it supplies many different ions that is important for the reverse transcriptase to function properly and optimally. So those ions include potassium and ammonium ions. We also have something known as magnesium chloride. It's a cofactor to make sure that the reverse transcriptase is functioning as it's supposed to be, and it can also boost its amplification. We can use magnesium chloride to make it work faster and produce more cDNA. And lastly, we have primers. So if you remember, primers are the short nucleotide sequence that binds to the poly A tail at the end of the RNA and initiate the reverse transcription. All right, okay, we're ready for the new part, qPCR. So master mix is what we need for qPCR. So inside the master mix, there are some ingredients in there. So the most important ingredient would be something called dye-based master mix. So it contains all the essentials for basically amplifying that cDNA. So it includes the buffers, enzymes, the NTPs that we just talked about in the previous slide. So these are the components for the qPCR. And what is different about this is that it got something called cybergreen. Cybergreen 1 is a fluorescent, and that is what we use to detect basically the amount of DNA that is being produced. We'll talk more about cybergreen later. The second thing would be primers. So reverse transcription, we see that primers are used to initiate that process. We also need primers for qPCR for the polymerization aspect. And what is different for these primers is that it's specific to the genome that we're interested in. We only want to polymerize the specific gene in that DNA sequence that we're interested in, because only that specific gene is the thing that we measured at the end of qPCR. And what is interesting about this is that we have a forward and reverse primers. So we have 2 primers for each gene. And lastly, we have autoclave water, so RNase-free water, and we use it to dilute the master mix to get our desired concentration and amounts. So this is approximately what master mix is made of. So the total will be 100%, so about 20% will be autoclaved water, 40% will be the dye-based master mix, and the other 40% will be primers. All right, so we just talked about primers. There are 2 different types. So why do we need 2 different types? So you have something called forward and reverse primers. So for primers, it synthesizes the upper strand using the bottom strand of the DNA as template, and then the reverse primer does the opposite. So it uses the upper strand as the template, and it is used to synthesize the bottom strand. So at the end, you get 2 strands of DNA, and since DNA is double-stranded, you need 2 different primers to synthesize the top and the bottom strand. All right, so we'll talk about setting up qPCR plates. So this 4x4 square is what we use for one gene. So each gene, we have 3 replicates. So we basically line them up. So sample 1, sample 2, sample 3. You put the replicates as where the samples goes. So no template control. What we do in there is that we add 5 microliters of RNase-free water inside the no template control. So the purpose of having this column is that just in case that there is some DNA sample or that's contaminated, or maybe the enzymes are not working, we can have this column to determine if there's any error that might be affecting the results of our samples. So we add, okay, I should also mention the controls. So after you put in the fly touchscreen, okay, after you put in the fly head sample, head 1, head 2, head 3 in its appropriate column, you also have the control and also the test flies. So you put the control flies at the top 2 row, and then the experimental fly in the bottom 2 row. And then RNase-free water inside the no template control. And then afterwards, we put in 50 microliters of master mix. So what is expected for the no template control is that there shouldn't be any values. So there shouldn't be any results in the no template control because there's no DNA in there. So enzyme shouldn't be polymerizing water. And there shouldn't be any positive results happening. If there is, that means that something went wrong in your experiments. So it is like a control to make sure that everything is going as accordingly and there's no other outside variables affecting your experiment. And so we have that 5 microliters of sample as well as 15 microliters of master mix. So the 5 microliters of sample, it would contain basically your DNA that is converted from RNA from the fruit fly, and then the 15 microliters of master mix, it contains the basically everything that's needed for polymerization to happen, as well as you have that fluorescent dye in there to measure like how much of that DNA is being produced. All right, so after you added 5 microliters of the sample, 50 microliters of the master mix, you want to put that qPCR plate into the centrifuge for 1 minute at 1,000 rounds per minute. So basically you want to eliminate, well, I think you mix the solution in there as well as possible. And then you put it in the qPCR machine, and the qPCR machine would basically have a thermal cycle that allows those DNA to amplify. So we'll talk about what happens in the qPCR machine. So these are the qPCR steps. So the first step would be denaturation. So it happens at 95 degrees. And what it does is that it separates those 2 DNA strands apart. Since DNA strands are double-stranded, you want to pull them apart so that the primers can bind in there to start the amplification process. And then after that, the qPCR machine will lower its temperature to 63 degrees Celsius. So DNA primers can finally attach since those double-stranded DNA strands have separated. And then it will maintain the temperature at 65 degrees, and that's when the DNA polymerase can start synthesizing and putting in those base pairs and make that new strand of DNA from your sample. All right, so we also talk about cybergreen is inside that master mix, dye-based master mix. So what this cybergreen does is a fluorescent dye. and it goes between the DNA base pairs. So when those, okay, yeah, so once the new DNA is being formed, as the new DNA is being formed, once those base pairs are being formed, that fluorescent dye will basically bind to that base pairs and release fluorescent signals. So why is this important? Because we measure the amount of fluorescence being produced by cybergreen, and measuring that fluorescent signal can allow us to determine the amount of double-stranded DNA present, so basically how much of that DNA is being amplified from our sample. All right, so we'll talk about what happens to the cybergreen fluorescent in qPCR. So in our first step, we have denaturation, so the separation of the double-stranded DNA. So when those double-stranded DNA separates, it allows the cybergreen to go between those two strands. And then when the polymerase are working to put those base pairs together, you have that cybergreen basically intercalating inside those two base pairs and releasing signals once it becomes double-stranded. So it will release the fluorescent signals as the double-stranded DNA forms. And then, and those fluorescent signals are also being recorded by camera during that process. And then lastly, we have the accumulation. So as time goes on, as the cycle repeats over and over again, you have more and more of the fluorescent signals being produced, and those signals will be captured and be used to determine how much DNA is present, how much DNA is newly produced inside the test tube. So we just summarize what we just talked about with qPCR, real-time qPCR. So what happens is that you have this DNA template that you start off with from your sample, and then you have basically undetected fluorescence. Okay, so it's not really undetected fluorescence intensity, but so when you add cybergreen into the solution, it will already produce small amounts of fluorescence. And then you start the qPCR step. So the first step will be denaturing that DNA, so separating those two strands, and then that allows the primers to bind, that allows the fluorescent screen to go between the two DNA strands, and then you have the primers binding to the DNA strand and starting the polymerization process with the polymerase. And as the polymerase creates new DNA, that cybergreen will bind to the base pairs that is newly formed and release fluorescent signal. All right, and then we'll talk about interpreting the results from qPCR experiments. So this is usually the graph that's being produced by qPCR. So as you can see, during qPCR, there's an accumulation of the DNA products, and those products are being visualized and detected by the fluorescence released from cybergreen, and then that reaction progression is being monitored after every cycle. So the more cycles that is being performed, the more fluorescent signals will be detected, and basically producing this shape. So we just talked about fluorescence accumulation. For cycle number, so you see this is a CT. It has a threshold with a gray line. So it is the cycle number is the number of cycles required for your sample to go from just the sample itself to enough DNA amplified to reach and surpass the threshold. So the amount of cycle for it to amplify enough DNA to reach that threshold would be called cycle number. Okay, so as you see that the green line doesn't start at the bottom of the graph, so it means that when cybergreen is being added into the solution, it already released the low amount of fluorescence. So there's already low amount of fluorescence being produced, even though the DNA haven't started amplifying yet, and the cybergreen hasn't binded to the newly formed DNA yet. All right, so we'll talk more about those three phases. So exponential phase, so it usually occurs in the early amplification cycles, and it generates the products in an exponential fashion since the qPCR enzymes are working at its top efficiency. The second phase will be linear phase. So as the PCR progresses, the efficiency is starting to decrease because there's lower amounts of consumables, like for example, the NDPs, primers, and enzyme degradations. And then the plateau phase, so basically at the end of the qPCR step, the product formation is greatly decreased because you get a depletion of consumables. Usually when we do qPCR, we're trying to compare between different genes. So usually it has multiple, sigmoidal curves. So this green curve right here, so since it reaches the threshold with less cycles, so the cycle value would be lower. So what it means is that since it only requires less amount of cycle to reach the threshold, it already has a higher amount of DNA inside the sample. Comparing to the blue curve right here, so it reaches the threshold later on with more cycles. So what that means is that it has less DNA to start with, so that means less RNA to start with in a sample, so therefore it requires more cycles before it reaches the threshold. And therefore you can compare between genes and be like, okay, for the gene that is represented by the green curve, it has a higher amount compared to the gene that is represented by the blue curve because it requires less cycle to reach the threshold. All right, so that's the end of the presentation, and I got just 5 questions for you. And the first question is, which of the following is not an ingredient in the master mix? So cDNA is like what your sample has. So if your master mix also has cDNA, that definitely contaminate your sample with other DNA sequences. Yep, so which phase has the maximum qPCR efficiency? Easy question. Okay. Which gene has a lower expression in the sample? Blue or green? Okay, that's the end.